Dear students, the aim of today's practical is to study the determination of LD50. This image will give you idea about what is meant by the LD50 and what is meant by the LC50. See, suppose that if 6 animals were there and if they are receiving any dose of the drug and that will resulted into the 50% death in the population of animal, that is called as the LD50. That means what? The lethal dose and the concentration at which the population is killed by 50% is called as LC50. The study of chemical or physical agents and their interactions with biological system to produce a response in an organism. The dose makes it the poison. So any type of the adverse effect of drug which resulted into the organ damage or lethality, that all the chemicals or physical agents, we can call it as the poison. Now there are two types of toxicities are there. One that is called as acute toxicity and the another one that is called as chronic toxicity. Acute toxicity, that is the amount of substance which is capable of causing an immediate reaction, that is toxic reaction in the animals or human beings within 24 hours. That is called as acute type of toxicity. What is chronic type of toxicity? The small amount which does not cause an immediate reactions but it accumulated into the body to cause a delayed reactions over the longer period of time, that is called as chronic type of toxicity. The toxicity test is need to do with the goal, that is select a test species that is good model of human, a response that is not subjective and can be consistently determined, and the test period that is relatively short. This testing will also give us the idea about dose response curve. The dose response term that gives us the idea about the toxic dose at the low concentrations. That is the lowest, or which is also called as TL10, that is the lowest dose introduced by any route except inhalation, reporting to produce any toxic effect in human beings or to produce the tumorigenic or reproductive effect into the animals. Toxic concentration at low, that is TC10. That is the lowest concentration of substance in air to which humans or animals have been exposed and has produced any toxic effect. Lethal dose, low, that is LD10. That is the lowest dose of substance introduced by any route except inhalations which has been reported to have caused death into human or animal beings. Lethal dose 50, that is LD50. This is the calculated dose of substance which is expected to cause the death of 50% of entire defined experimental populations. Lethal concentration 50, that is LC50. It is the calculated concentration of substance in air to which is exposed to cause the death of 50% of an entire defined experimental population. Using these response informations, one can compare LD50 of chemical gives a relative rating of toxicity and LD50 in milligram per kg and multiply by 70% gives a rough estimate of the toxic potential for the human beings. So now we will see in details the lethal dose 50. This is the amount of toxic agents. It may be a poison, it may be a virus, it may be a radiations that is sufficient to kill 50% of population of animal usually within the certain time. It also called as median lethal dose. expressed as milligram of substance per kilogram of body mass, compare the toxic potency of intensity of different chemicals, a measure of acute toxicity. So the lethal dose 50. So suppose that the one substance, that is, suppose it is A substance and another is it is B substance. A substance 10 milligram causes kidney toxicity and B substance 10 milligram causes neurotoxicity. So which is more toxic? We can't say. So for that purpose we have to do the LD50 calculations for both A and B, and if the A has calculated less concentration to kill the 50% populations, so obviously we can say that A is more lethal one. So what are the signs recorded during the acute toxicity studies? These are increased motor activity, anesthesia, tremors, arcing and rolling, clonic convulsions, tonic extensions, lacrimations, strobe reactions, salivations, muscle spasm, writhing, hyperesthesia, loss of writhing reflex, depressions, ataxia, stimulation, sedation, blanching, hypnosis, cyanosis as well as analgesia. What are the different methods for determination of LD50? The one that is graphical method, arithmetical method, statistical approaches. For research purpose, the most widely used method is Litchfield and Valaxon test. For routine practical classwork, read Munich, Miller-Trainer and Krebs methods. The arithmetic methods, that is Carver methods, graphical methods, that is Miller and Trainer method. So for calculating LD50 by any one method, we have to find out the least tolerance, that is smallest dose, and 100% mortality and most tolerated highest dose, that is 0% mortality by hit and trial method. Once these two doses are determined, select at least 5 doses in between them and observe mortality due to these doses. The percentage mortality value are converted to probate values by reading the corresponding probate units from the probate tables. The plot probate value against the log doses and read LD50 value as the dose that correspond to the probate. What are the probate values? These are the Blease proposed transforming the percentage kill into the probability unit. That is what we call as the probate. Probate table aids other researcher to convert their kill percentage to his probate, which they could then plot against the logarithm of the dose, and thereby it will hope obtain straight line. Such as so-called probate model is still important in toxicology as well as in other fields too. The graphical method of Miller-Trainer. The Miller-Trainer method is started used in getting LD50. The dose is plotted against the probate value. Based on the graph, LD50 will be estimated. The experiment demonstrates the determination of LD50 of neostigmine on the experimental animal and its comparison to standard LD50 of neostigmine. So this is for our understanding. We are taking one drug, that is neostigmine, and we are selecting out the different doses. For example, 0.1 milligram per kg, 0.2 milligram per kg, 0.4, 0.8, 1.6 and 3.2 milligram per kg, and which is given by intraperitoneal route. The toxical informations which is present for these are different by oral route, for subcutaneous, IV, by intramuscular, by intravenous, in mouse and rats and etc, etc. So these are different. So by using all these informations, we are choosing these different doses. So what are the objectives? The general objective is to determine the median lethal dose of neostigmine introducing in mice by intraperitoneal route. Specific objectives to determine the number of death per dose of neostigmine 1 hour after intraperitoneal administrations. They determine LD50 of the given dose by using Miller-Tenter methods. They compared experimentally determine LD50 and theoretical standards of LD50. So whatever the materials and methods used for the LD50, these are mostly used the mice. So we can choose out the different numbers of say mice over here. So here for this experiment 60 mice were used, which is approximately of same weight, and the neostigmine, that is 2.5 milligram per ml injections, is selected. So we can group them in different groups. So in each group 10 animals were there, and then in geometrically increment per milligram per kg, we are give the dosing to them. For example, we will start with the very lowest, that is 0.1, then 0.2, 0.4, 0.8, 1.6 and 3.2, like that. The number of dead mice within 1 hour was counted. Abnormal behavior, tremors, seizures observed need to be noted. What is the rationale for using the geometric methods? Because it is cost effective and allow the experimenter to see the effect without sacrificing the many animals. What is the results? We apply the correction factors, that is 0% and 100% mortality group. Percent response conversion to the probates. Convert doses to log dose. The probate value and the percentage of death against the log dose need to be plotted like this. So we are having such type of probate table in which the number of death and the percent of log dose is given. So we can have the computations like that. See, suppose that 20% response will be there. So if it is 20%, so what is the probit, that is 4.16 like this. So suppose that the response will be 2.5%. So in table there is no 2.5%. So what we can do? We can do adding the probit for 2 and 3 and divide them by 2, that will gives you the average for 2.5, isn't it? So that will be the probit of 2, that is 2.95, and for 3 that is 3.12, and we can divide them by 2, we will get the probit for 2.5% of response, and that will be 3.035 like this. So in this way we can calculate the we can plot the graph, we can have the probit units on that and log response over here as well as the percent response. So we can interpolate them to the 50%, so we will get the value. So from regressions we will get the probit log dose line, and the log dose was exploited corresponding to the probit at unit 5. The extrapolate dose corresponds to the median lethal dose, and antilog of these log dose will gives you the value of the LD50. And the dose corresponding to the 50% of probit 5 was taken as the LD50. So what are the limitations of this method? The LD50 gives you the measure of immediate and acute toxicity. Results may be vary greatly. LD50 is not tested on humans. All the relations to humans are only based on the guess. LD50 test is neither reliable nor useful because the human lethal dose is difficult to be predicted from the animal studies. Dear students, now we will see the multiple choice question for today's experiment. And what units are lethal dose 50 express? ounce by lb, milligram by lb, ounce per kg, milligram per kg. The correct answer is milligram per kg. Lethal dose 50 value are determined by amount of substance that kills at least 50 individuals of test population exposed to the substance, kills the test population when given at least 50 ml of this substance, kills the at least 50% of test population exposed to the substance, kills test populations when given at least 50 gram of substance. The correct answer is C, that it kills at least 50% of test population exposed to the substance. The concentration of toxin that will result in the loss of 50% of population called as LD50, IPM, TP50, none of these. Correct answer is LD50. LC50 value as means lethal dose 50, lethal concentration 50, both A and B, and none of these. The correct answer is lethal concentration 50. Toxicity is related with the instability, volume, temperature, doses. The correct answer is D, that is doses. LC50 associated with the toxicity studies. Yes, no, both A and B, none of these. The correct answer is yes. It is associated with the lethal concentration 50.